Glycogen storage disorders. So basically, glucose can be stored in form of glycogen. So we all know food contains glucose. For example, imagine this is a glucose molecule. After having food, these glucose molecules can enter into your GI tract, especially mentioned the glucose absorption happens in cases of intestines. So these glucose molecules can enter and get reabsorbed at this place. So we have a special receptor which helps in this absorption, that is SGLUT, that is sodium glucose uptake transport. We have two types actually, SGLUT1 and 2. SGLUT1 is present in the intestine, GI tract, and the other one is in proximal convoluted tubule, the kidney. We'll discuss more about these GLUT transporters in upcoming videos. So we will focus on the SGLT1 receptor. As I said, the glucose molecules from the intestine absorption happens into our circulation. For example, it enters into the bloodstream. So you can see the glucose molecules enter into the bloodstream, and thereby it increases the blood glucose level. So this blood glucose can reach to various organs and for its metabolic demand, as well as it requires stored form also. So the storage happen in cases of the liver. So this structure which I am showing now here, it's a liver. So glucose storage happening here, conversion to glycogen. As the receptor present in intestine, there is one more receptor, GLUT receptor, which is present on the surface of liver, that is called GLUT2 receptor. Actually, we have 5 types from GLUT 1, 2, 3, 4, 5. 1 to 5 we have. We'll discuss more about these receptors. For example, we'll discuss about insulin, whether the insulin dependent or insulin independent, where is its location, and whether it is active or passive. For example, GLUT2 receptor is present both in the liver and pancreas. So once our glucose molecule absorption into the liver, then there is a conversion of this glucose molecule into its product, that is glucose 6-phosphate. The enzyme responsible for this attachment of phosphate to the glucose is glucokinase and hexokinase. We don't have both enzymes in the specific liver. We only have glucokinase in liver and pancreas, and we have hexokinase in all other organs. So why we have these two different enzymes for a similar mechanism, similar pathway? So because glucokinase present in liver and pancreas, it cannot be inhibited by your the product, which is glucose 6-phosphate. In contrast, the hexose kinase can be inhibited by the higher amount of glucose 6-phosphate. So I'll teach you about this little further. Glucokinase, if there is high glucose in the blood, the product, which is glucose 6-phosphate, will be higher. So if glucose 6-phosphate get higher, it should not inhibit your glucokinase. Once it inhibits the glucokinase, there is no uptake of glucose into the liver. So it causes hyperglycemia. So we don't want that. So now this coming to this glucose 6-phosphate, it is the main byproduct of various, it is the main substrate for various other pathways. For example, glycolysis, Krebs cycle, oxidative phosphorylation for ATP synthesis, HMB, as well as cholesterol synthesis. Now we'll mainly focus upon the how glucose is glycogen is formation from the glucose. So this glucose 6-phosphate convert to glucose 1-phosphate, UTP glucose, and the final product is our glycogen. Actually, this is not the total final product. This glycogen which is forming here is a linear. Can you see here, each bead is one glucose. So it attached glucose in this manner, like linear manner. So it increases its area. So we don't want that. So to decrease it, to decrease the area and to increase the surface area of the given glycogen chain, we have something called as branched glycogen. So thereby the glucose molecule attached sideways like a branch. So the enzyme which is doing this branching is called as, name itself indicate, branching enzyme. But some patients, there is a deficiency of this branching enzyme can lead to a glycogen storage disorder, which is called as Anderson's disease. So in which there is accumulation of this linear glucose molecules within the liver. We'll discuss more about this glycogen storage disorders itself in part 2. So this branched glycogen now stored within the liver, and this process up to now from the glucose till this glycogen, we call it as glycogenesis, that is synthesis of glycogen. So for the breakdown of this glycogen, there is a first step in glycogen breakdown, that is the formation of limit dextrin. What is limit dextrin? It is a simple compound. It has 2 to 4 glucose molecules present as a branch. So this is the simplest structure of a glycogen. We call it as limit dextrin. The enzyme which is responsible for this conversion is glycogen phosphorylase. Once you don't have glycogen phosphorylase or it is deficient, it can lead to another disease, that is McArdle's. That is also one type of glycogen storage disease. Then this limit dextrin can either taken up by lysosome or still present within the cytoplasm for its further metabolism. So let's see what happens inside the lysosome. Once the limit dextrin taken up by the lysosome, there is a special enzyme present within the lysosome, that is alpha 1,4 glycosidase. This alpha 1,4 glycosidase convert and break down this limit dextrin into small molecules of glucose 6-phosphatase, and this glucose 6-phosphatase released to cytoplasm. Deficiency of this enzyme within lysosome leads to Pompe's disease, another glycogen storage disease. Hence, since it is a problem in the lysosome, we can also call it as lysosomal storage diseases. So as I said, this glucose 6-phosphate molecule released into cytoplasm for its release into systemic blood circulation. So that is with the lysosome. When the limit dextrin is present itself in the cytoplasm, then it can again further the branched glycogens, the branched glucose, that is 2 to 4 glucose, which I said before. Now that will be break down until there is only one glucose present as a branch. So we should now, this is not a limit dextrin anymore. So the enzyme which is debranching this, which is cutting these branches, we call it as debranching enzymes. So this is a opposite for the branching enzymes. We can call this enzyme, we can name these enzymes as debranching enzymes. Now, so we have 2 debranching enzymes. One debranching enzyme name is 4-alpha-D glucosidase, which metabolize limit dextrin to this product, and there is another enzyme which further break down this, the single glucose present as a branch, to convert into linear glucose. This is 1-alpha glycosidase. Sorry, I forgot to write that one. So it's actually 1,6 alpha glycosidase. So these enzymes will make the linear glycogen. Now this linear glycogen will break down into small glucose 6-phosphatase. If you don't have this debranching enzyme, then there is deficiency. There is no breakdown of limit dextrins and thereby accumulation, which can result in Cory's disease. This is another type of glycogen storage disease. But once in normal human beings, this metabolized linear glycogen now breakdowns into glucose 6-phosphatase. In this cytoplasmic glucose 6-phosphate from the lysosome and this cytoplasmic glucose 6-phosphate now should not enter into these pathways. So to prevent that, there is one enzyme which is taking back this glucose 6-phosphate into glucose by removing one phosphate at the 6th position. This enzyme is very important. It's a rate limiting step. So the Deficiency of this glucose 6-phosphate enzyme can lead to a very important and high-yield glycogen storage disorder. This is type 1 glycogen storage disorder, which is Von Wricht's disease. We'll discuss about this specific in detail in other videos. Now, this glucose 6-phosphate converted into glucose can enter outside the liver and enters into blood to increase the blood glucose level.